Was this enough?
Yeah, that's
great. Great. Thank you very much. So keep the mic and you go.
Good afternoon to everyone in the room and everyone joining us online. It's my pleasure to introduce the final plenary from the Guy Foundation sponsored section at this, my first QUEBS conference. And I want to say thank you to the organisers, especially Esther, and to all of you who have engaged in such fascinating and interesting conversations. It's been truly inspiring to come to such an interdisciplinary conference. Not something I've seen at such a scale, conversations between physicists, biologists, and everyone in between. I look forward to being a regular feature of these conferences. So in the Guy Foundation section, we've already heard from Dr. Travis Craddock on the first day, talking about spin beyond magnetoreception, and yesterday from Dr. Philip Curian, who spoke on life beyond photosynthesis. And we would like in this talk to talk to you about some of the projects that have been undertaken in the bioenergetics laboratories over in the UK on 2 different sites. But firstly, a quick introduction to the Guy Foundation for some of you who may not be so familiar. The Guy Foundation guides and facilitates exploration into quantum biology and the role it could play in advancing medicine, headed by our founder, Dr. Geoffrey Guy. So the Guy Foundation curates, leads and funds interdisciplinary research collaborations and has published a number of scientific papers as our outputs. It hosts an active network of 350 scientists coming from institutions and citizen scientists from across the globe. We also convene regular online symposia, the latest of which will be coming up in autumn, so please do sign up in time for the autumn series. And through these works, we hope to divulge not only the work inside the Guy Foundation, but topics that come up that we think are of interest from the wider community. In terms of resources, Dr. Geoffrey Guy has recently released his own book on quantum biology, A Glimpse into the Future of Medicine, which can be found through the websites, as well as links to our quarterly reviews, partially curated by the fantastic Bethany. And here you will find probably coming up soon, the feedback and our conclusions from this wonderful conference, as well as previous events and all of those research outputs. So finally, from this introduction, I'd like to invite you to scan our QR code. It can also be found on the website for the Guy Foundation or on LinkedIn, or you can even email the fantastic Nina, who will put you on the mailing list for the series coming up in autumn to attend our international webinars. And they have been, don't worry, they have been selected time-wise that it should work whichever hemisphere, country or university you seem to find yourself in at the time. Moving on to our work at the different sites across the UK, investigating quantum for health at Harwell. The first question you're probably asking there is, where on earth is Harwell? And I'm very willing to answer that one for you. So the research complex at Harwell is a central laser facility building over outside Oxford in the UK. Essentially, if you go to visit Rhys in London, you can jump off the plane, fall onto a train, and within about an hour, you'll be over at the central laser facility. So we're very close in space and even closer in terms of our research. So through the Guy Foundation Bioenergetics Laboratories, this has allowed us to collaborate closely on projects where we bring our unique specialities, different approaches to systems, and in some cases, it's even led to us building new bits of equipment for them to use over in Westminster. Something that I would like to talk you through is a small project that was undertaken a few years ago at Harwell looking into NADH. So we've heard a lot about cytochromes, we've heard about FADs, but something that Professor Stan Botchway, who I think a lot of you are familiar with, and he sends his regards, something Stan's been very interested in is NADH. So you may, of course, know that this is shuttled between NAD+ and NADH and is prominent throughout our cells and is essential in your mitochondria as part of the electron transport chain for the production of ATP. And as you can see along the bottom, the benefits of NAD+ have been hooked into by the slightly venomous supplement community, where you can now buy NAD+ as a gummy, as a tablet, as a pill, those are 2 distinct things, as a powder, or even as an injectable. But what's very interesting about this is that taking in NADH in these means has not actually been shown to uptake into your cells. So they've struck on something that is interesting, but they haven't quite worked out how to utilize it. So why are they so interested in NAD+? We're going to hone in specifically on NADH itself. And the question we were really asking is, does it work in a similar way to chlorophyll? It has a high electron yield, much like chlorophyll, and we've heard from several previous speakers about the interesting properties of the excitron passing through our extended system of chlorophylls to our reaction center. And actually, our mitochondria, including our NADH, work in a similar way. So with our quantum brains on, could this be an example of quantum tunneling in human biology? Can we form an analogy between these two systems? However, it is quite difficult to image a single molecule of NADH. I don't know if any of you have tried. So we have a slightly different workaround for that at Harwell. And as photophysical chemists, of course, we default to some photophysics. So NADH specifically is naturally fluorescent. It absorbs between 320 and 380 nanometers. And there is some thought that this could act as some kind of natural sunscreen. And it emits between 420 and 480 nanometers. It has a very low quantum yield. It's only 2%. But with that, we also get this massive quantum yield of photoionization of nearly 50%. And what's super interesting about NADH is that it has complex properties in solution, which we would not expect of such a small molecule freely diffusing. NADH has two excited states with distinct fluorescent lifetimes. One of these is shorter, around 300 picoseconds, and a longer one around 700 picoseconds. The community is still debating what the exact root of these two different components is, but it's generally thought to arise from changes in the local environment. The way we're investigating our fluorescence properties is to utilize time-correlated single photon counting. A few people have mentioned before, so we won't do too much detail. But specifically in our setup, we utilize our polarized pulsed light source, and we also use polarized matched detectors so that we can examine specifically the anisotropy coming through the system. A second added benefit to our system is that we also use multi-photon excitation. So in the case of NADH, instead of using one photon of 350 nanometers, which is not ideal when imaging a live cell solution, we can instead use two photons of around 700 nanometers, utilizing a quasi-state during the excitation with these two very closely arriving photons. So the two-photon absorption cross-section is an important property if we're going to utilize two-photon. And for NADH, this is very small. So we measure cross-section in units of Goppert-Mayer, named after the woman who discovered it. And at 750 nanometers, this is around 0.01 for NADH. For a dye, something like your fluoresceins or your rhodamines, we're looking at more at the tens of to the hundreds of units of GM. But we don't let that stop us in Harwell. Mostly because our two-photon has many advantages over one photon. We have better selectivity within our cell to reduce out-of-focus photobleaching, and we can also acquire higher anisotropy values because of our stricter set of selection rules. So the first step that was taken in these measurements was to look at just our isolated molecules of NADH freely diffusing through solution. When we take something like fluorescein under single photon excitation, we can see that we have a small but not insignificant anisotropy of less than 0.1. By contrast, when we take our NADH at the multiphoton, we are now getting an anisotropy of nearly 0.25. This is extremely high for a small molecule freely diffusing in a solution. We would expect our anisotropy values to be low so that when we hit our molecule with our polarized light, we expect it to be depolarized by the time it is again emitted. But we're not seeing that with NADH. And the trend continues when we add further adjustments to our solution of NADH. We can add FAD, sucralose, ethanol, even D2O, and we still see this very strong anisotropy value carried through the measurements. By comparison, a fluorescent protein, M-neon green, does also exhibit a very high anisotropy, but for a large protein, this is something we would expect. This is not surprising. So how can we further investigate the effects from NADH? First step to start honing in on this is to examine the effects of a restricted environment. So as I said, we already started with a fairly high anisotropy, 0.25. And as we start to add glycerol to this mixture, moving from 0 to 90% glycerol solution, we see further increases in the anisotropy. This is something we expect. We're restricting our molecule, we're stopping it tumbling. We will expect our molecule to keep that polarization. Moving towards imaging studies rather than spectroscopy, when we take our imaging setup, we're straight out using our excitation beam, which is polarized, no mirrors in the way, and directly collecting our emission afterwards. Moving over to a microscope, we're adding mirrors, we're changing directions, and every time we reflect that beam, we are changing our angle, our polarization of the beam. So what we need to do first is use something to ensure that we know the polarization of our system. And in this case, that M neon green that I told you about, it's possible to crystallize this protein. And in this way, we can show the polarization of our system. So in the white light image on the left, we can see there are in fact two crystals that look very similar in our white light image. When we move to the situation of vertical excitation and collect our emission also on the vertical plane, we see a higher intensity from our crystal at the top. So we can say that this crystal is aligned with our vertical direction. However, in the bottom image, we only really see emission from our larger lower sample. And this crystal is aligned in the horizontal direction with its emission on the vertical. Now we've characterized our system, we can move from free-floating NADH to some isolated mitochondria that have been taken from a cultured cell sample. So I don't know how well that's showing up, actually better than I expected. So each of these little white spots you can see, these are our isolated mitochondria, and each of these white spots gives us a decay profile that we would say is characteristic of our NADH decay. So in the vertical excitation and vertical emission case, we see a much higher emission intensity from this from this coordination. Twice as much intensity is collected in the VV direction as it is from the VH direction. Another way that we can look at this is through this 3D plot. So we can see not only are we getting more emission in the VV direction, but we are we're collecting emission from different mitochondria through these two different collection directions. So the peaks we see from the VV are not the same peaks that we're highlighting in the VH direction. Another view of our isolated mitochondria, just to highlight further that we've got this differential polarization. When we move instead from the vertical to the horizontal, we again see these uneven spots of excitation, and circled in blue are those mitochondria which are showing up in one image and not in the other when we are collecting with the other detector. The other combination of these collection and excitation directions with our vertical excitation, horizontal emission, and horizontal emission, horizontal excitation, vertical emission, we again see a different population of mitochondria emitting in these directions. So this is again evidence that we are seeing alignment of our NADH through the mitochondria, and we can selectively collect our emission from the ones that we are looking for. As we move towards a whole cell image, the system and the behavior was further characterized by looking at our intensities. So it's been calculated that we have between 80 micro and millimolar concentration of NADH in mitochondria. This is how we are still working around having our low quantum yield and our low cross-section. And we can also show that by exciting at 750, we are able to selectively excite the mitochondria structures, so we're not getting too much interference from other cellular features. One of the key questions here is, why are these mitochondria aligned, and what exactly are they aligned to? So on the left here, you can see one of my own FLIM images of some HACAT cells. And in this case, we are not polarizing our collection. We're simply collecting all of the emitted photons and building up one lovely picture. So you can see with the shorter average lifetime, we are seeing more shorter average lifetime from our NADH populations in the nucleus. But what we're really looking at is the cytoplasm with our mitochondria. This is where we are seeing those longer average lifetimes and much higher intensities. In this case, we used MCF7 cells to examine the anisotropy of our NADH in these live cells. And from these two different views, comparing the VV with the VH in both cases, we see that the anisotropy between these two channels is between 0.3 and 0.4, so up towards the higher value of what we can calculate, with an upper limit being a roughly 0.55 for anisotropy values. So this multi-photon excitation shows that we have got unequal emission in the vertical and horizontal channels. And we have this range of values according to the orientation of our NADH within the mitochondria. At this point, I would forgive you for walking away from this lecture thinking that all of your mitochondria are all pointing in the same direction. They are all looking north. But that is not always the case. So some of the cell samples that were examined, they did not have the highest emission intensity from our VV channel, but instead from our HH channel. So in this case, we've got more intensity from, we are exciting more NADH in the horizontal direction, and they are emitting in the horizontal direction. We are postulating that this could be evidence that as long as there is alignment within that sample, it's not necessary that every cell, every sample needs to be vertically polarized, but that as long as there is agreement within the system, that is what is important for the passing on of this electron behavior. And to further highlight the point, we have those contrary directions. So that's our vertical excitation with our horizontal emission, and again, our horizontal emission with our vertical excitation. And as we expect from the system that we've shown does show this anisotropy behavior, these are much dimmer. We see far less emission from these systems than we did in our lovely bright VV and HH cases. So to really hammer this home, we see this as possible evidence of preferred mitochondrial alignment in living cells. And while fluorescence is not the best way to look at this, it has been a valuable tool that has been a first step towards this journey of understanding how this high photoionization output could be behaving in cells. In conclusion, we've shown that NADH polarization is quite high in standard solution, and it's not fully explained by what we know about the structure. When we restrict the environment of NADH, this anisotropy rises greatly in glycerol. So this indicates significant influence from the environment, such as that mitochondrial structure. And with a forward look, something we have been doing in Harwell is looking to build a system for examining with entangled photons, as was mentioned by Alix and Alexandra. So as another Alix, apparently we all have a love of entangled photons, and hopefully we can apply that understanding to this system and further directly interrogate the photoionization rather than the fluorescence characteristics. So we're going to move towards performing photon correlations and to measure the effects of isolated mitochondria and photoionization in an entangled photon setup. And we want to examine evidence of a quantum mechanism for NADH via some simulation studies. Before I hand over to Rhys, I just want to give a quick thank you for everyone who worked on that project specifically to Holly Smith, who carried out all of these measurements over in Harwell. Thank you so much for listening, and I will pass you over to Rhys.
Yeah, thanks, Alix. Afternoon. My name's Rhys Mould. I'm a postdoc at the University of Westminster. And I'm going to talk about the work we're doing at Westminster, supported by the Gaia Foundation, which has taken the shape of two areas: ultra-weak photon emission and hypomagnetic field exposure. And I think we could summarize what we do at Westminster as perturbing or exploring areas where there's quantum effects in biology with the tools that we have as cell biologists. So let's start with ultra-weak photon emission, or UPE, also sometimes controversially known as biophotons. So UPE refers to the passive emission of light from living cells. When I say passive, I mean that there's no direct input of energy. So your cells are constantly emitting this light because it occurs as a byproduct of respiration, and specifically the production of reactive oxygen species, or ROS. So it's unlike any direct input of energy like in fluorescence or bioluminescence. So your metabolic processes in the cell produce excited intermediates, and when those relax to their ground state, they do so releasing a photon. So the best studied model for us is the mitochondria, the powerhouse of the cell. This diagram shows the electron transport chain, which turns metabolic fuels into ATP. At sites or complexes 1 and 3, you have the production of ROS. And you can either have the direct production of a photon through singlet oxygen production, or in interactions between ROS and nearby lipid molecules, which cause lipid peroxidation. These produce excited states, and when they decay, they produce a photon of light. So in essence, it's a form of bioluminescence, except that there's no direct enzymatic pathway, such as the luciferase pathway, and it's orders of magnitude less bright or less intense. And there's some interesting ideas about how this light that we produce can interact or propagate within a cell. For example, there's this idea that the optical properties of the mitochondria themselves, which aren't just the punctate little beans you see in medical or biological textbooks, but instead can form long elongated networks that propagate throughout the cell. Perhaps these biophotons or this UPE can propagate through the cell in the mitochondria, acting as like an optical waveguides or an optical net that spread, or fiber optic nets that spread throughout the cell. And Philip mentioned this yesterday, that the production of UV photons could interact with nearby tubulin in the microtubule network. And these resultant excitons can propagate through the cell and potentially mediate cellular signaling. So what's UPE for? Well, it's quite a contentious area. So there's this idea that it's a vent for the cell to dissipate excess energy. Or for the last 100 years, there's this idea that it may facilitate non-chemical communication between cells or organelles within those cells. That includes us, which we published a few years ago. The problem is that the data on UPE is relatively sparse compared to the data talking about UPE. And this is because it's a technically challenging, at least for cell biologists, to study. The challenges are that the UPE intensity is extremely low. We're talking like a single to 100 photons per centimeter squared per second. And there are competing light emitting processes that occur within a cell, which if you don't differentiate between the two can lead to confusion between the two terms and where the light's coming from, and so on. So this is delayed luminescence, in which a material can absorb light and then re-emit that light after that exposure is taken away. Sort of like how your skin will radiate heat after you walk in from standing in the sun for a while. So we need to detect UPE, we need a system that is both sensitive enough to detect single to 100 photons from a sample, and that has completely eradicated any background light because it will just wash out the signal by orders of magnitude. So that's what our colleague Alistair did at Harwell. He built us this, what he calls the beam machine. So it consists of a single PMT counting head. That's in a black aluminium foil sheet. That's in a dark wood box, and that's covered by a blackout cloth. That's kept in a dark room in the basement of my university, and that's separated from other rooms by another dark corridor. So multiple levels of dark proofing, but it's necessary. It's also useful to put as many doors as you can between people wandering in and blinding your PMT. So Alistair really cut the teeth of this this apparatus on mung beans, which are beans that will happily grow in the dark. So he plates beans, you stick it under the detector, and then you can see on the plot that the number of photons coming off of the sample increases over time, which is both a consequence of the beans growing and them becoming stressed as they consume the water in which they were originally plated. So Alistair designed this separate setup that we now can take into Westminster to look at mammalian cell culture, which is my bread and butter. So I mentioned delayed luminescence, and I wanted to illustrate how much of a difference the sample preparation can make when we're trying to detect these very low levels of light. So if you expose your sample to ambient light, that's just the light that's present in the lab day to day, and then you put it straight into the detector, you end up with this large exponential decrease in light spiking and decaying over the course of 2 hours compared to samples protected from light. This occurs even in cell-free media. So you don't even need a biological sample for this to occur. It even occurs when you have an empty dish. So the polymer that we use, the polymer dish that we use, can also absorb light and also will spit it out, which can lead to conflation between UPE, as we're trying to study it from a biological sample, and this physical process, which has nothing to do with the metabolism. So take all into account, and I take a population of cells. I take 500,000 cells and I seed them onto Bailie dishes. I leave them overnight in the dark. Then I put them in the incubator. It's the simplest experiment in the world. But you can see that you can detect light coming off of your cells, the intensity of which increases over time compared to the media. This is MCF7, a breast cancer cell line. You see a similar trend in A549, which is a lung cancer cell line. You also see the emission from non-cancerous breast cell lines. The emission is less intense, which is interesting because I think one of the things we want to be able to use this for is as a marker of stress or perhaps redox status or cancer status, as a label-free measure of those things. So the emission of the intensity of emission is meant to be linked to the production of ROS. So if you increase ROS, which is easy to do in cell biology, you should see a subsequent increase in the number of photons you're counting. Makes sense? So the objective here is to manipulate UPE using the drugs that we have in the lab. So I was able to, we doubled our output and have a second PMT with a second photon counter. So we can do more tightly controlled experiments. We stuck in a load of doxorubicin, which is a very potent anti-cancer drug, and compared it to a vehicle-controlled DMSO. And sure enough, in MCF10A cells, you can see this very large increase in the number of photons counted when you treat the cells with a stress-inducing agent compared to the control. So far, we've been able to distinguish UPE from delayed luminescence. We've demonstrated robust evidence of UPE from a single-layered mammalian cell culture. And we've also seen that you can increase the intensity of UPE by increasing the amount of stress in the system you're measuring. There's loads more work to do. I think what's most important for us is to be able to get a spectra of these emissions. That's fraught with its own challenges itself. But that would tell us if you can differentiate between different cell types, whether they're cancerous or not, whether some stresses induce increases in emission at certain wavelengths over others. And that's something that we're actively working on. So let's move on to part 2, hypomagnetic field exposure. So the Earth has a geomagnetic field of low strength, about 56 microtesla on average. Most known for protecting us from harmful solar radiation, but it's also been an ever-present factor throughout the origins and the evolution of life. You may have heard about the radical pair mechanism, but what I was interested with this is that if your biology is sensitive to weak magnetic fields, such as the GMF, what happens if you take it away? It turns out quite a lot. So this is a review by Sarimov, I think it was 2023. And in vivo, I don't know how well it's showing up. There's lots of unexpected effects and also contradictory effects in different models. So there's in planaria, there's increases and decreases in their proliferation. One of the interesting ones is an increase in the number of mistakes. I don't know what the context was for that. I didn't read that one. In vitro, which is what I'm interested in, there's changes in morphology, also dependent on the cell line you're measuring, changes in cell differentiation, changes in calcium expression, and so on. So there's this plethora of effects that you get from just removing your cells from the geomagnetic field. And the context that we're interested in looking at this is in space, because we like it or not, the Artemis mission is set up to facilitate long distance space travel. That's either to Mars or long duration space travel with a permanent place on the moon. And this is phase 2, which specifically highlights that. And it's our experience that the consequences of removing a magnetic field from astronauts is sort of flown under the radar in terms of how they're going to respond in terms of their health. So to study this in vitro, we are using mu metal chambers. Mu metal is an alloy of high permeability, which means it blocks or deflects low frequency magnetic fields, sort of like a Faraday cage, but for low frequency fields. And we've been looking at two cellular models, THP1s, which are an immune cell, this monocyte specifically, and C2C12, which is a muscle cell. The muscle cell is quite interesting because there's already plenty of data suggesting that, well, that spaceflight negatively affects your muscle. So let's start with the THP1 cells. Normally, immune cells will respond to lipopolysaccharide treatment, LPS, which is produced by bacteria, with an increase in ROS. But what we found was that response was muted in cells treated or kept in the hypomagnetic field for 24 hours. We also noticed visually that these THP1 cells can differentiate. You can change them from a monocyte to a macrophage. And as they do so, they are initially a suspension cell, so they float around in solution. When they differentiate, they become adherent, so they'll stick to the surface of whatever you're growing them in. And we found that after 4 hours, you can increase the rate at which they become adherent. So they seem to be differentiating from monocytes to macrophages faster. And we also found that markers, cellular surface markers associated with adherence are significantly increased in response to being in the shield for 24 hours. And finally, in THP1 cells, we looked at the arrangement of actin. So in these images, the nucleus is stained in blue, the actin is in green, and these are the cells treated with the control or just kept in physiological conditions. And in the cells kept in the shield, you seem to see this derangement of the actin arrangement pointed out by the arrows. So there's quite a distinct change in the actin cytoskeleton network in these cells. And this is, I think this is 24 hours. So moving on to muscle cells. So to start with the muscle cells, we were interested in their rates of proliferation following a wound. So in cell biology, this is a simple assay. You take a population of cells, you slice down the middle, and then you measure microscopically how fast it takes for those cells to close their gap. And indeed, we found that cells treated over 16, 24, and 48 hours recover that wound healing process much slower than the cells kept in the control. And next, so Victoria mentioned the seahorse yesterday. So the seahorse is, it's not a seahorse, it measures mitochondrial function in real time. It's pretty much the gold standard for mitochondrial function assays in cell biology. And what we found is that with muscle cells treated with the hypermagnetic field for 24 hours, you see a significant decrease in mitochondrial function as a result of this. I should mention that for any cell biologist, this was in the, so T2C12s are either myoblasts or myotubes. We looked at myoblasts to start with because it's easier. So to sum up, we found that hypermagnetic field exposure affects immune cell stress responses and the rates of differentiation. We also found that hypermagnetic field exposure decreases muscle cell proliferation and mitochondrial function. I want to take the muscle cell work further and probe the specific complexes where we think it might be occurring. And there's literature, well, there's discussion in the literature about how it's exerting its effect. Is it a radical pair mechanism or the radical species? We don't know. Or is it to do with the iron sulfur clusters within complex 1? And this is all work that we would like to undertake. So with that, I think I've sped through this, but I'd like to thank everyone at the University of Westminster and the Central Laser Facility, including Alix. Thank you, Alix. Our colleagues at the Molecular Foundry, the Lawrence Berkeley Lab, and of course, the Gaia Foundation, who made the conference